Okay, then. I think I'll just start to warm us up a little bit, just to sort of, I think we're just about to get started now. Hello, folks. Welcome to YR. This is the second YR webinar of 2021. And joining us today is Professor Susan Holmes from Stanford University. And also we're joined by my co-host here, who's joining us from Finland, Dr. Leo Lahti. I let Professor Holmes introduce herself as part of her in the presentation, and later on at the Q&A, I will be joined with Dr. Leo Lahti, and we'll sort of lead the Q&A for today's session. Thank you very much for joining us both, by the way. Also joining me is the producer here, Marcin Kozinski, who is a technical whiz kid, and without him, YR definitely owes him a great debt of gratitude for his quick thinking. I will sort of start off here by handing over immediately, actually, and just to sort of hand over to Dr. Susan Holmes. So I'll just sign out. Or just sort of stop my video and hand over to Susan Holmes. If you're joining us in YouTube, feel free to put your questions in the YouTube channel and we'll keep an eye on them. But also just actually let us know where you're from, because we're very interested in our worldwide audience. I'm here in Ireland, we have North America here, Poland and Finland, but we're joined from people all over the world, and it's always good to see that, actually. So Professor Holmes, I'll let you take it away there.
Okay, thank you so much for having me. Actually, I'm not in North America, I'm in Portugal. But I'm happy to join you here anyway and talk to you about why I use R to analyze the human microbiome. And so I'm a statistician and I work in collaboration with a lot of biologists. And I've actually been using R since 1998, when it only existed on Linux machines. But the microbiome project I've been working on, we've only been doing this work for about 17 years. So it's not the beginning. I was already using R. But the kind of data that we have is homogeneous data. And we could say that actually homogeneous data are easy because you may just make matrices. And the big challenge for us has been moving to heterogeneous data. And heterogeneous data are all heterogeneous in their own way. So we have different ways in which we see that. And in the human microbiome, I study mostly the effect of antibiotics, also diet perturbations, clean out, and study pregnancy. So we have a lot of really important medical data, and we want to keep all the aspects of the data together. And I'm going to explain to you some of the challenges that we have. So the data are heterogeneous, and I'll show you in detail what that means on a case study. The challenges which have to do with the data structures are that many of the data structures are not actually matrices of real numbers, but we have a lot of graphs and a lot of trees, and I'll explain where they come from. We also have very high dimensional data and what we call multi-domain or multi-omics data. So we have structured high dimensionality. And we want to do things like testing this multi-domain data using a graph structure. And then it's really important to have something which is reproducible. So the heterogeneity, let's start with that. The heterogeneity comes both from the fact that sometimes we're doing supervised, the example of pregnancy where you see the length of the pregnancy is the response, or unsupervised. And in that case, we don't have a response that we're trying to predict. So that's a heterogeneity in the status of the variables. But there's also technical variation in the different data types. So you could have continuous data or categorical data or graphs and trees, images, spatial information. So all of that gives for things which are not on the same scales, not measured with the same types of measurements. So that's a really difficult type of heterogeneity to deal with. On the other hand, as a statistician, you know, if you took an early statistics class, you see that people study what's called IID, so independent and identically distributed. Our type of data are never like that. So we have time series, we look at longitudinal study, sometimes we have spatial data, but they're all very dependent. So what is the microbiome? It was defined by Josh Lederberg as the ecological community of commensal and symbiotic and pathogenic microorganisms that literally share our body space and have been all but ignored as determinants of health and disease. So this was about 20 years ago that people realized that we had this huge amount of organisms which made our ecosystem healthy or not. And so we have genes in the genomes that we can measure with next generation sequencing. And the amount of actual bacteria that there are, the number of, is almost at the same order of magnitude as the number of cells in the body. So it's a really important component of us as symbiotic organisms. And we have the metagenome is the composition of all the genes that are present in those different species, and they're all doing different things. Now, when it comes to actually knowing what's being produced, you can measure either by gene expression or by metabolics, and I'll show examples of that. But you mustn't think that the bacteria are necessarily pathological. Most of the time, the bacteria have a function which is really important. And I would say that right now, with what's going on with COVID, the microbiome in the nose and in the respiratory tract is really something which is the sort of first line of encountering in the ecology. So these bacteria are with us all the time and part of our contact to the exterior. So the data themselves, and if you're interested in genomics and you know that in some sense there's the DNA and the RNA. So the DNA is just what's there, and the RNA, and in particular the messenger RNA, is going to tell us what's turned on or what could potentially be turned on. So the genetic signature that we are very interested in is that of one particular gene, which is different in every different strain or species of bacteria. And this gene is called the 16S rRNA gene. And then we have a lot of other types of information. We have proteomics, so we have chemical compounds that are present in the samples that we take. And we also have clinical information about the patients, their age, their body weight, their clinical status. We might also know what part of the world they come from, what they eat, what season it is, when this is taken. And then we have a lot of exterior knowledge, contiguous knowledge about the domain. And so we know about the evolutionary tree of the bacteria that are present. We know about the metabolic networks of the genes which are present. And all of that has to be recorded and digitized and incorporated in the data analysis. For me, as a statistician in the way I work, I don't think of anything as metadata. Everything for me is just data, and everything is really important. So here is a diagram from a science article from about 10 years ago, where we have the phylogenetic trees, that is the evolutionary relationships between different species. And here, these little red dots shows the presence of certain of these species on the tree in different locations in the body. So this is a little diagram in some sense of what the data are. And we actually work with people from many disciplines. And we could say that ecology meets the clinic in the study of the human microbiome. And so we have the immune history of the patients and clinical variables at the same time as we know something about the genetics of the host, the person. And we have a lot of bioinformatics sources of databases and different formats. And we have to take into account that what we're studying is both proteomics and metabolic pathways are involved, but also that we are studying an environment. So we use a lot of methods that come from ecology. And you'll see, multivariate methods, measures of diversity are standard in the study of ecology in general, and it also applies to the microbiome. And then we have methods in statistics. So in statistics, we decompose variability and do studies which take into account dependencies. And this is one of the big reasons for using R is there are a whole lot of packages already available for studying how to decompose variability. The other side of the microbiome is the study of the evolutionary relationships and the dynamics of evolution. And we also have some elements of network science. So we're studying microbial communities, which are in networks, and gene interactions, which are networks, metabolic pathways, which are networks. And we have very high dimensional data, which we have to visualize. So we do a lot of dimension reduction, and many of this is done using mathematics. So the way we conceived as a structure or an environment for working with these data was to use R to import data of different types. And so this is the heterogeneity I was talking to you about. That is, we know a phylogenetic relationship between the different bacteria. We have taxonomic tables, and the different species occur in different counts. So this OTU table for us, statistician, it would be just a table of counts like a contingency table. So we have to put all of this together. And we made a package called phyloseq, which was a package for dealing with the fact that we had all of these heterogeneous components. But in particular, there were no packages available for combining phylogenetic trees and networks with contingency tables and enabling plotting in ggplot at the time when we did this. So this work was done more than 10 years ago, and the second publication is the one that we did in PLOS ONE, where we published all the different ways of using phyloseq for analyzing the microbiome. So the trees and networks, for instance, we have even an interactive version of phyloseq, which works with Shiny, where you can go through and, for instance, make a network, and we can use a special threshold for deciding whether we connect together taxa according to whether they co-occur. And in this picture, you see this is practically a clique of different taxa, which when they're present, they're all present together. And this is a useful thing to know. That is, you have a very tight-knit community of bacteria which are co-occurring. So what's the phylogenetic part? And this was sort of where I came in from and why I got interested in this type of data. I've been interested in phylogenetic trees for a very long time, and especially in their encoding in programs like R. So how do you build it? Well, we start off with aligned sequences. In this case, we have different types of malaria parasite. So Falciparum is one of them, but we have many, many different types that come from. So Falciparum is this one, 4, but we have ones that come from many different hosts. And you want to find out the evolutionary relationship between them. And recently, of course, you've seen a lot of trees that came out of Trevor Bedford's group, where you see the tree, the evolutionary relationships of viruses, and it's done for COVID, but it's the same type of output. That is, you have a phylogenetic tree, and you can see which are the most recent common ancestors, which are close together on the tree, and so on. So this is one representation of a phylogenetic tree. And we build the tree. And we can consider it's like it's unknown, and we build it from sequences, and we build it in different ways. I won't go into the details, but we have many different ways of estimating this parameter. And what was great was there's already a package available in R for doing this manipulation of phylogenetic trees. Emmanuel Paradis is in Montpellier and in the south of France, and he developed this package a while ago. And now we have even better packages which are built on the structure that he used, ggtree and Fangorn. And so I'm going to show you an example of how, in some sense, there are bad ways and good ways for manipulating tree data. And in this case, for instance, the standard way of doing it for the biologist was to use what was called the Newick format, which is a bracket format for the different groupings in the tree. And you see there are lots of brackets, but one of the problems of this representation is it's very sensitive. If you're missing an open bracket or a closing bracket, your expression, your representation won't pass. And so you're going to have an error. So it's a really clunky format, which was originally developed by a group of biologists around Felsenstein. And there's actually a much better way of representing this. And that's the way that APE does it. And that's through the use of a matching. And so in this case, we have this group clade, we could say 9 and 10 are a group because they're sibling pairs. And so you just put those together. And in this case, 11 and 6 is also a group. But then what happens to the ones that are not, that don't have tip labels? So here there are 11 tips. And so the number 12 is assigned to the first available, the youngest child. So in this case, we'd have 12, which is assigned to 1 and 4. And here we have 2, which is grouped together with 11 and 6. So 11 and 6 is actually assigned the number 13. And then you have 2 and 13 as a grouping, and then that goes on. So there's actually several ways of building bijections between what we call here matchings and phylogenetic trees. And I'm not going to go into the details. You can look, wrote a paper on this more than 20 years ago on this way of coding. And then I discussed it with Emmanuel and said, you know, this would be a great way to encode it in R. And this is the way it's encoded in R. And I'll show you, we can come back to that, but this is the details of that. Now, apart from the phylogenetic tree, which is slightly trickier actually than networks, in networks it's very simple how to encode them in R. If they're sparse, you just want to be recording the edges. And that's the way it's done in one of the main packages which already exists called iGraph, and other packages which have been built on iGraph in R, ggnetwork, ggraph, and so on. So where do these occur in microbiome data? Well, it's co-occurrence networks. That is, if you have 2 samples and they have a lot in common in terms of bacteria which are present, which are in common, then you want to make an edge between those 2 samples to say that they're similar. Or you could do the dual, that is, you could take 2 taxa, and if these 2 taxa co-occur in many of the samples, you also make an edge. So there are different types of what we'd call a threshold graph. So they're geometric graphs defined by a threshold, and as I showed you, the co-occurrence is the main criteria. So we use similarities and in some senses also weighted graphs. And we have different ways of using these similarities, and I'll come back to them. We don't, in general, for this type of data, use correlation networks because it's sensitive to the depth at which the sequencing is done. And the correlation coefficient only looks at linear relationships, and the co-occurrence is often nonlinear. So the sparsity and nonlinear make it better to use what we call a Jacquard index. So the Jacquard index, for instance, it's in many of the ecological packages. The main one I like is called vegan. And the Jacquard index was actually introduced for this looking at instances of plants in the alpine meadows. And Jacquard decided that you want to take into account, in some sense, only the species that were present in one of the 2 places that you were comparing, and you don't want to take into account the species which are absent together. So if things are absent together, they would make the correlation bigger. because it was a joint zero, but that's actually artificial and biases the study, especially in the case when there are many, many zeros. So Jacquard index is better. And again, vegan is a great package for doing environmental studies. And so this is just an example of how you compute in R the Jacquard distance between two presence-absence matrices, so 2, 0, 1 matrices. And we use this a lot for doing the bipartite graph, for instance, between samples and taxa. So this Jacquard index is used in a sort of more mathematical way or machine learning way. It's a positive definite kernel, and it's used for comparing graphs. You can decide that you look at presence absence of edges and two different graphs on the same nodes, and you can use a Jacquard similarity between them to do that. So it's a good way of looking at that. Now here's some output that comes from microbiome data, where we have Jacquard co-occurrence graph. And so the numbers here are samples. They have to do with patients. And so the patient number is the one that we put on the node. And then we colored them according to whether these are patients, pregnant patients, whether they had their babies preterm or not. And when we make a network like this, which is quite easy to do in R, either using ggraph or ggnetwork, you can see that just by looking at the data here, these are all grouped together. This means that all of these samples, which come from the same patient, they share bacteria in common, and they're different than those of other patients. Now, of course, anybody who studies the microbiome knows this. That is, there's a huge subject effect. That is that the differences between people are the major sources of variability in a sample. But this is a way where you could see this pretty well. And so here we have the samples, but we do have some sharing between samples. And then you can do a test, and I'll come back to that, as to whether the red and the blue are separate or whether they're sort of mixed in this at the same level. But this is built as what we call a Jacquard index is computed, and a threshold is taken, and if the distance is not too big from a Jacquard viewpoint, then we put an edge in the graph. And then this is done at the level of the taxa. That is what we call the dual graph. That is, it's a graph not on the samples, on the taxa themselves. And if the taxa are present in several samples together, then we see that they'd be, you'd see an edge. And so these taxa have just been numbered. And this is done with ggnetwork. So in this case, you might be able to see communities or you might not. This is quite complex, and you'd have to have a representation maybe which is a bit more sparse than this if you want to see them. So to come back to where I was, I gave you a couple of examples. For the microbiome data, we need phylogenetic trees. So we had APE, a package. We have BIGAN, which was a good source for ecological indices and distances. And then, of course, we have ggplot. That is, as soon as we're going to do high quality plotting, it seemed important to be able to ggplot. When we wrote Phyloseq, actually, in 2010, we started working on it. ggplot was only just starting, and there were no bioconductor packages, for instance, which use ggplot. Nowadays, everybody knows how to use ggplot, but the way we started off, ggplot seemed obscure to many people, so we just wrote a lot of things as wrappers. So the plot ordination is just one function, which is a wrapper around a particular kind of multivariate plot. And we try to make it as transparent as possible. That is, the people who started using Phyloseq didn't know ggplot, and many of them didn't know R. So there was a lot of trying to make things as easy as possible and defining a lot of things by default. So here, this is a plot network case where it's the same kind of network that I just talked about, where we have a threshold on the Jacquard distance, and we do a lot of things by default so that you don't have to choose. And here's the phylogenetic tree with the different sample prevalences along the phylogenetic tree. So it's pretty easy to make high quality plots with ggplot, and you can even make contour plots and things like that. So that's the goal, was to have heterogeneous data, very high quality plots. Now, how do you combine the different data types? That is, I talked to you about the phylogenetic tree. I talked to you about the networks that come in. And we actually have functional networks, and sometimes we look at things across time. And usually when we're trying to summarize the data, you have to use some kind of multivariate, multi-domain method. And the workhorse for much of the ecologists, at least, is to use some version of multidimensional scaling or principal coordinate analysis. And this is, you know, an old method, which comes from a mathematical method from Schoenberg from 1935. But it allows you to take data which has, you know, 2,000, 5,000 dimensions and bring it down to 2 or 3. And it's based on a singular value decomposition in its standard instance. And in that case, we like using fancy versions of multidimensional scaling, which have been made available within the package ADE4, which is a package developed by ecologists in Lyon. And so those packages allow to do things like take into account the distance along a tree when you're trying to plot samples. So that's called double principal coordinate analysis. And I'm not going to go into, I'll give you a link for the slides, but Elizabeth Purdom, in her thesis and also in a paper in Science that she published with David Relman and his team, they use this method of double principal coordinate analysis, where they map the distances, but they take into account the distance between the different species, and you can plot them. So the study that she was interested in was an antibiotic time course study, in which you have 3 different patients, but you make them take 2 time courses of antibiotics, and you take many samples along time. And we're going to look at different taxa, and the biologists call them OTUs. And we look at them across 50 time points. And we want to look at what happens before they have the antibiotics, during the first course of antibiotics, and between the antibiotic time. So here, these are the way double principal coordinate analysis works, is you do a first multidimensional scaling, or first principal coordinate analysis, with a distance between the species as it's seen on the tree. Then you take a low dimensional representation of that, and you take centers of gravity, which are given by the weights of the different taxa for each of the samples. So this gives you another representation. You do another PCOA. And here you see that we have the 3 different patients are grouped differently. So we have the blue patient, which are very grouped here. The red are quite diffuse, and the green just occur along this gradient. And we can also interpret the left to right in a sort of biplot way. We have the different taxa here. And again, this is something that is due to using tools that come from R, that were developed by the team in Lyon, and applying them to the case of the microbiome. So it really was a case where the use of R sort of inspired and enabled us. And here we have the main difference that we wanted to look at, which was the under antibiotic stress was 2. And for the 3 different patients, so we have D1 and D2, E1 and E2 are different, but we see that the third patient is highly resilient, didn't have a big change between when they took antibiotics and when they didn't. So if I want to follow the communities over time, instead of just looking at under stress or not under stress, we can also do that. And we did this with a very simple Markovian model. And so this was data on pregnancy that I mentioned before, where we're trying to find out whether there's a relationship between preterm birth and the microbiome. And we took actually samples at very many body sites. The one I'm going to talk to you about is the vaginal microbiome. And in that data set, we had 49 women. We took 40 women we did the training on, and we had a validation set of 9 women that we had set aside. We made predictions, which we checked on the second set. So this was done through making a Markov chain of states. That is what happened if you look at the microbiome. And I'm just going to show you a plot first. I'm going to make a co-occurrence network between the different samples. So just take this picture. Each column here represents a sample, and there are many samples per patient. There are between 10 and 15 samples per patient. And then we look at the co-occurrence of the samples. What do they share in terms of bacteria? And you see that this first group here, we've grouped them together in what one could call in a sort of stochastic block model type representation. We see this very highly red block. So all of these samples share a lot. All of these samples share a lot. So we have 1, 2, 3, 4 very distinct blocks. And here there's no real distinct block. That is, the sharing it's very diverse. And what's important is we have preterm up here, which represents whether or not the patients had their babies early. And we see that mostly in the fourth group, that's where most of the preterm birth is occurring. There is a little bit of preterm birth in the first group, but mostly it's in the fourth group. So we knew we were mostly interested in this group. And this is done in some sense by just looking at heat maps and co-occurrence networks and trying to understand. And then we look at some of the questions that we came up were, when are these alterations occurring? And I won't go into too many of the details of the scientific paper, but all of these plots were enabled by the fact that, there are nice packages, nice tools available. So for instance, pHeatmap is one of them. And so these heat maps allow you to look at the data and understand. So now I've represented the different bacteria in the samples. And here you see that the lactobacillus sort of explain the first group. And this fourth group, which have a lot of preterm birth in it, well, they don't have 1 or 2 particular bacteria. And so this is a case where it counts as the standard knowledge in ecology or environmental science, where you say diversity is healthy. This is a case where diversity is pathological. You have very high diversity here, but it's actually linked to preterm birth. So that's an example of a real data set where we made a lot of progress by looking at the data in this way. And then we made a Markov chain to try to understand the passage in and out of this sort of bad state 4. And we have a much better understanding of this unstable state 4. And again, this was made with a R package Markov, which is specialized in these Markovian models. And you can look at all the online material. I put some links here, and I'll put up the presentation that you'll be able to go in and you can see all the R code. So we published in the supplement the R Markdown and the data, so you can go over each step of this analysis and do it yourself. So the conclusions we had is we actually found, in some sense, some bacteria, in particular Gardnerella, which we went back and studied again in much more detail because it's highly associated to preterm birth. Now, the last thing I want to just cover very, very quickly, and I'll give you a pointer to my book, which I wrote with Wolfgang Huber on modern statistics for modern biology. And there's a whole chapter about graphs and networks. But I made this, it's about HIV. And we can do very, very similar things with COVID. Each of these points represent a patient and a particular strain of HIV. And then what we created was the minimum spanning tree. That is a tree which minimizes the distance in terms of DNA. And then we mapped it onto a world map. So you can see that there's a lot of travel. That is, Taiwan to Australia, China, even to Brazil, China to the US, and so on. And so, of course, we published this 4 years ago, but the same thing is occurring with COVID. That is the travel of the species, and you can track them with their distance in terms of DNA. And one can test the relationship between a graph, which is built as a co-occurrence graph, for instance, and a factor. So I'll just point you to this chapter here. We have the HTML, which appears for this chapter, where we have all the code that would enable you to reconstruct a graph. I had said earlier on, one of the things that we wanted to find out when you have a co-occurrence graph is, so here we have the same mice and then the mouse is the color, and then we have two different litters. And one of the things that we wanted to find out is, well, the mice, of course, there's a subject effect, so they're very similar to themselves. But is there a letter effect? That is, here we have round and round. Are there more edges which are round then there are edges where you have a round one on one side and a triangle on the other. So in order to test that, we have what's called the Friedman-Rafsky test, which is a permutation test where you permute the labels and you try and find how many hybrid edges there are. And so this is a graph-based two-sample test. In one case, you could do a minimum spanning tree, but you can also do a co-occurrence graph. And you can find out by permuting the labels whether the number of hybrid pairs that you get is exceptional or not. So in this example here, we did one with a minimum spanning tree, and you can see the actual value on the graph here, and the permutation distribution is over here. So you can see that the p-value is very small. That is the chance of getting something that's so big. And then you can do conditional tests. I want to give you time for questions, so I am going to wrap up, and I'll leave some of the slides where I won't make too many comments on, in particular about the Bayesian nonparametrics. And I'll just finish off talking about reproducibility and, again, YR. So the things that I already talked to you about were the phylogenetic tree and the ecology and making maps and using ADE4. One of the things I haven't talked about, which is really important, have to do with annotation of genes and manipulating strings. And that's where bioconductor comes in. That is, we use a lot of their complex S4 structures for what we call multi-assay experiments, where you have many different domains that you're putting together, and you have to have lists which have many containers. So bioconductor is a very interesting framework for doing that. All of the studies that I've shown you have been published with supplements written as R Markdown. And I always publish the R Markdown with the papers. And it's not very well written. It might be that the code isn't perfect. We try and make it as clean as possible, but we do publish our code at the same time as the paper. So we can't spend too much time making it the best we can. But it does allow other people to reproduce all the studies that we do. And I think R Markdown is a wonderful way of doing that. So that's the last reason why R, bioconductor, R Markdown. So there are many more things that I could have talked about, and I really don't have time. I started a little bit late, but I'm not going to go through all of these. One of them that I like a lot about the modern way of using R, and in particular the tools developed by the R Studio, is to use Shiny. And there are a couple of examples here which were developed by my students or collaborators. So we had a Shiny version of Phyloseq. Julia has a very nice program for doing adaptive generalized PCA. We can choose interactively coefficient. And then Chris Sankaran has a way of looking at longitudinal data with a tree. He actually uses D3 directly. It's not a Shiny app, but it's very interactive. So I really think that's the future. So what we have available right now, as I said, are ways of doing interactive graph visualization, ways of testing and representing graphs. We can use hotspot detection and conjoint analysis, and we have very good reproducibility and a reproducible research workflow where we can go all the way from the raw data, the sequences and the qualities, all the way through to an output of an analysis that you can publish. And so if you want to see some examples of workflow, we published one in F1000 Research with all the code. And at this point, I'd like to say that I think the reason why it's so easy to use and develop and then do research with R is because of the community. And so there's a very strong R and bioconductor community, in particular, of course, Wolfgang Huber, who's my co-author on the book, but his team who developed DSEC2. But I'm thankful to Emmanuel Paradis, who developed APE for doing phylogenetic analysis. And of course, the team at our studio, Hadley Wickham and JJ Allaire and Joe Cheng, who developed Shiny, is really, they've been wonderful tools to use. And if you want to see some of the codes and some of the examples, I put some links here. And then I want to thank my collaborators and my Elizabeth Purdom, who did her PhD, who was one of my first students to do microbiome data. And then my lab group, and Joey McMurdy in particular, who developed Phyloseq, and Ben Callahan, who's here, who developed Data2. And here we have Julia Fukuyama and Chris Sankaran, who developed the interactive tools. So I think I'll stop here. And then if you ask me for more details about something that I glossed over very quickly, but it would be good to have a discussion. Although I'm not going to open YouTube, so you'll have to, Leo will have to do the monitoring the questions and asking them for me. Okay, thank you.
That's great. I just that you unshared. Oh no, actually leave it up the screen. Leo, okay. I'd like to introduce Dr. Leo Lahti, who's joining us from Finland, and he's going to be leading the Q&A here. Essentially, I'll help him out here. But Leo, I'll let you take over there just to get started. I'll monitor the YouTube just to make sure that as more questions come in, that I'll give you a heads up.
All right, thank you very much. Very nice talk, Susan. And there are a couple of questions. Some are more technical related to R, and some are more towards the substance you were talking about. And the first one is for some of the listeners. Apparently, the bioconductor is not known. So there is a question: what is bioconductor?
Okay, so bioconductor is a sort of high quality version of CRAN for biological data. And bioconductor is a project which was started by Rob Gentleman, who wrote R, and jointly, quite quickly, Wolfgang Huber and a group of others, Sandrine Dudois, quite a few people worked with him on it. And the idea of bioconductor was to have data structures which were tailored towards genomics, and also to have curated packages. So packages in CRAN, they're tested by a computer, and they're run through 3 or 4 different operating systems. Bioconductor, it's not the same, and they're humanly curated. And there's a sort of certificate of quality in bioconductor packages. And it has helped the community because in the biomedical sciences, it's very important to have a package of high quality, which would give results which could be certified in some sense. So bioconductor has been going on for almost 20 years. And it's definitely something which I'm going to make a mistake. I think there are about 2,000 bioconductor packages, and they're maintained on a bioconductor website. And so sometimes when you call up a package, say on, it used to be, oh, it won't find the package because it's a bioconductor package. So there used to be quite a separation. Nowadays with our studio, we try to meld it. And so there's what's called a BioC manager, and you can install them very easily. A lot of heavy listing is done by the bioconductor, the permanent staff, because the goal is to have a lot of annotation. So there are lots of bridges in bioconductor between databases, which have ontologies and different annotations, and tools which put you in contact with meaningful biological interpretation. So those of us who do bioinformatics and genomics, bioconductor is our main tool. And so and it also has a sort of certificate of quality. So if you're looking for something which does, and it's in bioconductor, you can be sure that that package is going to be maintained, that it's been carefully curated. And so it's a very strong part of the genomics community.
Yeah, thanks very much. There is actually a related question from Dasala about this. Sounds awesome that you can share results of paper as R Markdown file. So is this some kind of industry standard that the papers can be shared this way?
Well, I don't know about industry standard. I mean, that's what I was saying about the fact that we work quite hard at making our R Markdown available as supplementary material in our papers. But sometimes we have journals who don't understand R Markdown files are text, as in Markdown, and they're only some journals, medical journals, where you're only allowed Excel or PDF. They don't have a structure for just a text file. So there are no industry standards, and we're still working at trying to get a lot of a buy-in from different journals from different areas. Now, bioconductor was very successful for genomics, and it's recognized by genome biology, by a lot of genomics journals. But the equivalent, in some sense, has started to happen in neuroscience, where they have something called Neuroconductor, and open journal way. They have various journals where they want you to submit either an R Markdown or a Python notebook or something like that, where people can reproduce what you're doing. And I think it's really important, because I didn't show this, but there's another talk I give where I show that in a particular study that was done, which was published in Nature, if you look at all the choices that were made along the paper, there are probably about 200 million different possible analyses that could have been done. That is, it's the forking paths, in some sense we call that. They have so many choices that combinatorially you have of the order of 200 million possible choices. Well, you can't use something like Benjamin Hofberg or something like that in order to correct for all of those possibilities. So what are you going to do to validate? That is, you can't compute a p-value and say that it's meaningful. And so what happens is you have this option of saying, okay, I'm going to show you all my code. I'll show you everything I did. And then you can run it and you can make a small change in my choice. That is, you could choose a different transformation, or you could choose to leave out a different set of observations. Suppose I left out 5 observations. You can see whether that has a huge effect on the outcome. Now, of course, it's going to have an effect, so the results won't be exactly the same. But qualitatively, you can look and see whether you get something which is equivalent, in which case you could think that the result is pretty robust. And by publishing your code, you can find out whether somebody worked really, really hard to just find a sweet spot for their analysis, or whether, you know, the choices they made were pretty standard. So it's really showing your plumbing, I mean, in some sense. And that's why I said, well, you know, it's always improvable. It's never perfect, but at some point, it's better to publish something than something perfect, as long as it's publishable. And so we do take the time to publish our code and make it available with the papers. And we've got a lot of useful feedback that people ask us, you know, why did you make this choice? Why don't you do this? And so it engages others to have a discussion around the code.
Yeah, we find it very useful. And I think the tools for doing this are also becoming more widely adopted, and people are getting more used to this. So maybe it's becoming more and more standard. It's also a generational thing, I think. So what are the biggest gaps in the R ecosystem at the moment for the microbiome analytics? Are there some clear shortcomings or things to improve?
So, I mean, so for the microbiome, for the tools that we try to develop, I mean, the big, I think for us, certainly in the studies that we're doing, it's the multi-assay experiment containers, that is having not only phylogenetic trees and the sequences, but the data that we collect are multi-domain. So you're also collecting information, for instance, about the host. So it's the microbiome plus looking at cytoph, or, you know, the immune profile at the same time as you look at the bacterial type. And you also have the possibility of looking at the genetic profile. So you have these different sources of data that you want to integrate. So the data integration and combining the data, I think that's the big challenge that we're all coming at with different angles. So people have, depending on where they come from, they have different ways of understanding that. But I think we all agree that that's the big challenge moving forward, of the data integration of all how rich it is. What I talked about was counts of 16S data. I hardly talked about the metagenomic data. And so we also can gather huge amounts of reads. And for the metagenomic data, we don't have very good tools for denoising and visualizing, because it's so much more complex. Because you have 2 domains, they come from many different genes instead of just one gene, and then many different species or strains instead of just a clear bijection between the 16S and the ASV. So.
Yeah, right, yes. I have seen there are some nice solutions for this in bioconductor, at least for this multi-assay thing. Then about this preterm data, there was a question on if the profile of the preterms depends exactly on how premature they were.
Yes, it's true that we made a 0-1 variable, preterm, not preterm, out of a variable which is original, a response variable, which was the gestational time. And so you can be much more refined but in some sense, the clinicians really care about the problem of preterm as a 0-1 thing. And I agree, but we did see very high correlation between this, what we call this community state type 4, where you had very high diversity and very low lactobacillus, and short pregnancies. So that's definitely something, and it's something we're still working on. We work with the Gates Foundation, and they're actually trying to find practical solutions which have to do with trying to alter in an interventionist way, trying to alter the microbiome. So there's some hope that that actually will lead to a translation into a medical solution.
Yeah, right. So if somebody wants to repeat this analysis, is it possible to access Phyloseq datasets on Bioconductor?
Yes, so Phyloseq is a package that you can download in R. And as many R packages, they have a lot of data already living in them, but also several of the studies I talked about, the PNAS study, all of those studies, the data are available as Phyloseq objects, and you can just download them and redo the analysis. So all of that is available. And in particular, the workflow code as well has lots of examples and lots of code. And so all of that is reproducible. Joey McMurdy maintains a very nice set of tutorials. And so that's why I point to his page, because this page has a wonderful set of examples and tutorials and material to go through.
Right. That question was from Eugene Hickey, actually, by the way. So thanks, Eugene, for this question. Then there is a question from Chris English about data versus metadata. So you referred to that in the talk. So what's your view of metadata?
Yeah, so I don't like, and I'll tell you a little bit more why I'm allergic to the use of the word metadata, because many of the databases maintained, say, by the NIH and things like that, they say, we made the data available, and they make certain sequences available, and then they don't make the information about the patients or about the subjects available. And they say, oh, we don't make that available, that's the metadata, and we're going to hide that. But in fact, if you don't know the clinical characteristics of the subjects, or you don't know their age, or they don't know their BMI, you can't do any kind of cross-correlation or studying association. So for me, that data is data. It's not metadata, and I need to have all the data combined. And that's why I like Bioconductor so much, because it glues all of this together. That is, within a bioconductor object, you have this sample information, which is glued to the other information. So it's in a table, you have a list of tables, but they're all kept together. And when you take out a few subjects, you just take out all the information about those subjects. You take a subset, and it's all done sort of automatically. So the manipulation of the metadata is done as if it were data. So I think it's really important to realize that there's only one thing, it's data. We have the data.
Yeah, sort of easy to agree with that. Susana Fuentes is asking, how about causal inference with microbiome data?
Yeah, so that's a very interesting question. Some of the studies that we've done are longitudinal. The only way I know to do a causal inference for microbiome data is to design experiments where you have the same subjects who have to go through interventions, perturbation interventions, because the subject-to-subject effect is so large and there's so much heterogeneity. that you can only do it as a longitudinal perturbation analysis. So we have done some causality, that we can do it, for instance, for the antibiotics, but that it's only feasible through, I think, through a clever design. I don't know how to do it computationally because there's too much variability that you don't control. So perturbation experiments enable you to establish causality. Now, of course, that there are some things that you can't do as a perturbation experiment, and you can't change, people's habits. And often, even about diet, people aren't so honest about what they eat or not, or they don't remember. So there are lots of things which are very complicated in doing perturbation analysis for the microbiome. And so,
Yeah.
But
Yeah.
it's the holy grail. So everybody wants causality, but it's much harder than we'd like.
Yeah, I guess so. Yeah, thanks so much. There is, I think, as many questions as I asked already, and it would be lovely to continue this discussion, but I think we are out of time now, and we just probably have to close here, but hopefully there will be further chances to continue.
Okay, thank you very much for having me. It's great. Thanks a lot.
I think Kevin still is.
Yeah, thanks very much. I'd like to just sort of close the session and sort of thank Professor Susan Holmes and Dr. Leo Latti for the great Q&A there. Thanks very much for that. I'm really happy about how this presentation turned out, because a little bit of a confession, I made a bit of a technical problem at the start, but we recovered very well. And so thank you again, and hopefully we'll all see you soon. Just to sort of close and say that we are, just two things we were going to say. Usually we are on Thursdays at 7 o'clock, but because our studio conference is on next week at Thursday, we're going to move to Monday. And we have a presentation about Power BI with R. So that's on next Monday. Then we'll back to Thursdays as normal. The second thing, actually, and again, something I forgot to do at the start, is to thank our sponsors, Jumping Rivers, who are great supporters of the R community. So that's it. Hopefully we'll all see you again in the future. And thank you for the everybody's contribution of their time and their enthusiasm in helping bring the YR community forward and the R community together in a really tough year over the last 12 months. So I think I'll hand over there to Marchand, our producer, to send us off.
Thank you. Thank you, Kevin. Thanks, Leo.